5, 4, 3, 2, 1, 0. All engine running. Liftoff. We have a liftoff.
Mini brains in space. Did you know that we can study something called brain organoids, effectively mini brains, in the special conditions in space on the International Space Station? And that such studies may help us with diseases like Alzheimer's, Autism, or Parkinsons. And there's actually even more than that. My guest this week is Professor Alysson Muotri from the University of California, San Diego. He and his team have flown such brain organoids to the ISS many times already. Don't miss this episode on what I personally consider one of the most exciting intersections of space and biology, and ultimately, hopefully, a source of potential massive positive impact right here on Earth. Enjoy. Hey, space enthusiasts, welcome back to another episode of the Space Business Podcast. And it's another episode in our mini-series on the intersection of space and biology, which I'm personally very excited about. And I'm super excited because today we really have a very well-known researcher on the intersection of space and biology, Professor Alysson Muotri from the University of California at San Diego. Welcome, Alysson.
Thank you. Thank you for having me.
It is a pleasure. And why don't we start off as always, if you can just give a quick introduction of who you are, what you're doing, that would be great for our listeners.
All right. I am a faculty here at UC San Diego in the departments of pediatrics and cellular molecular medicine. And also relevant for this audience, I'm also the director of the iScore, which is a novel center that we create here at UCSD to deal with stem cells in orbit. And so what my lab does is to use stem cells to model the nervous system. So we create what we call brain organoids with neurotissue derived from human pluripotent stem cells that mimic several aspects of human neurodevelopment. And we have been very successfully on using this tool to better understanding and treat neurological conditions such as autism, epilepsy, and other types of rare syndromes.
Excellent. So since our audience is probably not super well versed in biology, we're gonna unpack a little bit of the things you said there. So I guess the interesting thing to ask is what you described there, the brain organoids. There's probably many of our listeners who have never heard the term organoid before and exactly what that is. You mentioned that you're starting from stem cells, and I also want to take a step back there because I found in my own work with space and biology, there's still some misconceptions around from, I think they're going back to the 1990s. People hear stem cells and they get very scared because they're thinking embryonical stem cells. And maybe you can just explain what are brain organoids and how do we actually produce them today?
Yeah, no, that's a great point. And so let me step back and actually tell what kind of stem cells we use to do that. As you pointed out, there are different types of stem cells. The stem cells that we use are called pluripotent stem cells. Pluripotent because they have the capability to specialize in different tissues. And most of these pluripotent stem cells can be found in the embryo, in the human embryo. So the human embryo has these pluripotent stem cells while develops and forms the different tissues. And back in the 90s, scientists were harvesting these embryos to isolate the stem cells and grow the stem cells in vitro in the lab. Since 2006, 2007, there is a new revolution in this field called cellular reprogramming that was discovered by Shinya Yamanaka, who ended up winning the Nobel Prize for his discovery. And what he shows us is that you no longer need human embryos to do that. You can actually reprogram cells from live people. So this means that I can take any cell type from your body, a piece of your skin, your hair, your dental pulp from your gingiva, and I can isolate these cells in the lab and reprogram them to behave like human pluripotent stem cells. Because they can capture your genome, these are your stem cells. And what my lab does is to guide these stem cells to specialize into the brain cells. So some people are interested in liver, so they can guide the cells towards the liver. Other people are interested on the pancreas, so they can guide the cells towards the pancreas. We are neuroscientists, so we want to make brain pieces, brain tissue from these pluripotent stem cells. So we use a little bit of knowledge that we gain from embryology to identify the factors that we can add in our culture, in our petri dishes with these pluripotent cells, so they can start making brain cells, making neurons. And if you do this in suspension, the cells will self-organize. They will create like a three-dimensional structure and start producing brain cells by its own. So it's all genetically encoded. So we only kickstart the process and they do the rest. So as a consequence, we end up with some spheres. These are three-dimensional spheres that contain many cell types, similarly organized as the human brain. And these cell types produces like a really nice reductionist model for us to start understanding the interplay of the different cell types that we have in the brain. And again, because I can do that with people with neurological conditions, I can compare and contrast both of them to see what's wrong, what's different. For example, from a brain organoid derived from someone with autism or Alzheimer's. Do I see some of the alterations in these brain cells that helps me to explain the disease? And if so, can I actually use that to find a therapeutic agent or a strategy that can be used to improve their quality of life?
So if I understand you correctly, what you would do is you would create these brain organoids, maybe one that's a control, that's healthy tissue, and then another one that's diseased, so to say, with the condition you want to study. And you mentioned like autism, but there's obviously many brain conditions, Alzheimer's, Parkinson's, multiple sclerosis, and so forth. Is that what you would do? You have a healthy model and a diseased model, and then you compare what's going on there?
That's exactly it. And for most of the time, we can use diseases that have like a high genetic risk factor, Alzheimer's, autism, or we can actually study diseases that might be caused by an environmental factor, for example, the Zika virus or the coronavirus. So we were among the first to actually show that the Zika virus, for example, can cause microcephaly in the brain because it kills the progenitor cells that were migrating to form your cortex. We also applied that to the coronavirus at very early stages of COVID-19, and we predicted the neurological symptoms before the clinicians actually realized that COVID-19 also have a neurological defect. So allows you to study known conditions, but also predicted what environmental factors might actually create or impact your brain.
And so this sounds very important because that then seems to imply that, as with every model, I guess, and this is a model, one would have to worry that, well, maybe there's something missing that it really translates into the full organism, like the full human being that has the brain inside the head rather than in the petri dish. But I guess for your results seem to suggest that the spherical thing in the petri dish actually does model the brain correctly and translates into nature, so to say.
Yeah, so the model does a good job into conditions that are part of the neurodevelopment. And even though, as I mentioned before, it's a reductionist model, we don't have the same number of cells as the human brain, right? The brain has 100 billion cells. We have something that is 2 or 3 million cells in there. And of course, I mean, these neurotissues are not connected to sensory information. They're not vascularized. So there are still many aspects of the model that provides limitations to a better understanding. But the beauty is that this model can improve, and that's what science has been doing over the years. We are improving the model. Soon we're going to have like a model that contains vascularization, so we'll be able to grow like a bigger tissue. We are already like connecting these organoids with sensory information, vision, a spinal cord, or a painful receptor, so we can start adding that. But one important thing is even being like such a reductionist model is already giving us insights into the biology of these conditions. So that's why it's important. So it doesn't fully recapitulate, but like any other model, I mean, it has like advantages and disadvantages.
So what you're doing there with growing these mini brains in the petri dish, that already sounds incredibly sophisticated. But of course, we're a space podcast, and so we have to kind of now talk about space because what you have been doing for a while now is actually taking these models into space So of course the first question is why? Why
Yeah,
that because adding space to everything makes everything more complex and
right. Yeah.
expensive and time consuming and everything. So how did that come about?
Yeah, so that's an interesting question, and the reason is also interesting. So as I mentioned before, the model recapitulates early stages of neurodevelopment. So it's almost like we are reconstructing the brain of the person in parallel as an avatar inside the lab, and that starts from the beginning. And as you know, human development is quite slow. It takes like 9 months gestation. And plus, when you are born, you still require some time to mature. So the organoid model follows that. So because it's all genetically pre-programmed, it follows the same timing as the human. So if you want to study autism, that's a great model because autism starts in uterus. So before you are born, so we have the right tool, the right model to do that. Zika virus also starts in uterus. So the Zika virus crossed the placenta from the mom and infect the fetus. But now, if you want to study dementia or Alzheimer's or ALS, these are late diseases onset, usually age related. And you
Hmm.
need to age a lot the model. So the brain needs to be quite old for you to start seeing the symptoms, right? You don't have Alzheimer's when you are 10 years old. You have Alzheimer's when you are 60, 70, 80
Hmm.
years old. So it's unpractical for us to age the brain organoids for that long. So
Hmm.
I was looking for methods to accelerated aging in my lab so I could study that. And that's when I came across some literature from NASA and other investigators who actually cultivate human cells in at the International Space Station, or even cells from the astronauts returning to Earth after a period in space. And they all converge into the same idea that the space environment ages the cells. And I'm calling it space environment because there are multiple factors here that we don't understand yet. Is it microgravity? Is it cosmic radiation?
Yeah.
Is it magnetism at the International Space Station that is causing that? We don't know. So I'm broadly calling that space environment. So whatever happen in space, when you bring the cells back home on Earth, we observe this accelerated aging. And this can be measured by several molecular tools. For example, DNA methylation or gene expression, the amount of receptors. So we can use all those tools to kind of more or less guess the age of the cells compared to the ground control here. And when we do that, we realize that yes, the cell seems to be aging faster in space. So our first mission, when we first have like the proof of concept, was in 2019. And since then, almost every year we are sending like a mission to kind of confirm the observations that we have, and always adding a little bit of complexity. So that was, I think we finished like a 8 mission, and we have another one by the end of the month now, just to continue our experiments.
So okay, so I have like several follow-up questions on that. This is so interesting. So before we were going to space, like you said, with some of these conditions like Alzheimer's, you'd have to wait in a human life 50, 60 years most of the time. How did people study that? Were they just using like shorter-lived animal models, like mice who are living much shorter time frame? But then of course mice brains are not human brains. Is that what was happening before?
Yes, yes, and you touch like a very difficult problem in science, how to study these late onset diseases if we don't have good models. So the animal models that most people use is the mice that usually last like 2 years in the lab, no longer than that. So we don't have like an aging model. You could study monkeys or chimpanzees, but not only there is an ethical problem that start to emerge by
Mm.
using non-human primates, but also the how expensive it is
Yes.
to do that. So it becomes non-practical. So the model systems that we have to study these diseases do not recapitulate the aging part.
And And so you mentioned that we found that this aging process in humans, as seen in the brain organoids, is accelerated in space. Do do we actually have any idea why that's happening?
So we are trying to isolate the factors. So that these are the newest missions where we try to prove or disprove that certain factors are important. Of course, for example, microgravity. This is something that you cannot mimic here on Earth. Some people might even claim that, oh, we have like a machine that mimics microgravity. No, it does not. It changes the vector of gravity, but mimicking microgravity is impossible on Earth. So this is one factor that we cannot do anything about it. But for example, space radiation, we could shield our experiment, or even better, we are creating like brain organoids that are resistant to cosmic radiation by cloning inside genes from a species that can survive cosmic radiation. So that allows us to isolate radiation as one of the agents. But honestly, I think it's a combination of all these factors. I think that would be the reality. And if we are right, there is no other way to perform these studies except for in space.
Okay, so it sounds like there's a lot more research to be done, lot more potential PhDs for, researchers who want to still earn their PhDs. But one reason I'm asking is, and maybe I'm sort of like dreaming in a utopian way here, is if we understand why it accelerates in space, maybe in some way, thinking about longevity research, we could also understand how to slow it down on Earth. But I don't know whether we've done any research in that direction.
Yeah, that's also part of our research, how to rejuvenate or even deaccelerate the aging process. So absolutely, I mean, there are a couple of senolytic molecules that might be used for that. And there are new molecules that we are curating from the Amazonian rainforest biodiversity.
Wow.
That we have strong evidence that they might be neuroprotective and they might actually help your brain to age healthy. So we are testing those molecules as well.
And so, by the way, this fact that we've seen the accelerated aging in the space conditions with organoids, do we have any way of like verifying, for example, whether the same thing happens to astronauts? Has this been studied?
Unfortunately, again, we are reaching like a very sensitive point, which is the inaccessibility of the human brain. There is no way we can have like the consent of the astronaut to grab a piece of their brain to study in the lab, right? So we don't have tools that have the high resolution for us to study the human brain. And that's why using the brain organoids is kind of a proxy. But we can study blood, we can study skin cells. So these are tissues that are available to us that we can study. And if we do that, if we study these tissues in astronauts, you do see similar things. Of course, I mean, different tissues might respond in different way. But for the brain, the only way to do it is by growing brain cells in space.
Okay, I was thinking, okay, I'm not a biology expert, and I know there's also the problem of the blood-brain barrier and everything, but is there something like maybe like the spinal fluid? But I guess, then I'm advocating we should take like spinal taps of astronauts, and they probably won't be very happy with that.
Yeah, yeah. So I mean, studying like this very specific population, it is a challenge. Most of the time, because we know who they are, there are HIPAA protection. So this is like confidential agreements. For example, their data should not be released. So it becomes complicated. It's getting a little bit better now with private missions, where some of the private astronauts are consenting to study, and we are seeing like more and more publications on that. But the NASA trained astronauts is a little bit more complicated because they have all this protection. So yeah, so spinal fluid is definitely something that could be like a proxy. But again, I mean, we can look into synapses. We can look into how these neurons communicate to each other, what kind of neurotransmitters they are producing. There is so much more information than just looking to what's released into the cerebral fluid.
Gotcha. And so how many times has your lab gone to space now? And what have been some of the results you found exciting and noteworthy?
Yeah, so we completed 8 missions. We have the 9th mission now this year. First of all, I mean, there's lots of failure. We always talk about the positive data, but we made lots of mistakes and we fail a lot. So this is important for people who wants to do this type of research. I can say now that I have like a 10-year know-how. But when we started, there was lots of silly mistakes that we made. But going back to what we learned, so what we learned is that, yeah, the brain cells, they age faster after a period in space. Aging faster, it's hard to quantify because we also realize that this is not linear. We even coin like a new term because it is such an aging in a compressed time that we never see anything like that here on Earth. We are calling this space-induced neurosenescence or SINS to reflect that this is like a space unique opportunity or a space unique phenomenon that we don't see on Earth. And these brain cells, there are different types of brain cells. You have neurons, you have your glia cells, you have neuroprogenitor cells. Each one of these populations react in a different way. For example, the glia cells, mainly the astrocytes, they become quite reactive. So they are responsive to stress and they respond in a pro-inflammatory way. So we see increase in inflammation in brain cells that are exposed in space. Neurons like to make synapses, and if we just follow the trajectory of the cortical networks here on Earth, you see that they build in complexity until they reach like the higher complexity of the adult brain. When they return back from space and we measure these networks, they are completely different. So instead of following the same trajectory as you'd expect for someone maturing or aging, they already show signs of disruption of microcircuitries. So this would be something similar to what we see in dementia when people start losing the synapses and losing the circuitries associated with memory, for example. So we learned that as well. We also send, this is like a great example. In one of our missions, in two missions actually, we send brain organoids derived from Rett syndrome, RETT. Rett syndrome is a severe neurological condition where the kids are born normal for the first year or so, and then they start losing everything that they learn, all the milestones. They start losing the ability to talk, to speak, and then start losing the ability to walk or eat themselves. And they will end up a little bit later in life in a wheelchair with not much we can do. So I study Rett syndrome for, I think, over 20 years now. And I never heard about a neuroinflammatory component for Rett syndrome because, again, we studied the mouse model, and the mouse model doesn't last that long and doesn't have all the cell types that the human brain has. But once we send the Rett syndrome brain organoids to space, we noticed that, as I mentioned, there is a response, a neuroinflammatory response. And that was exacerbated into the disease condition. And then we started studying how come this neuroinflammation is exacerbated by Rett syndrome. And we figured out that it's because in Rett syndrome cells, they are losing like a protein that controls the expression of endogenous retroviruses. Your genome is full of endogenous retroviruses that accumulates throughout evolution. And we keep them silenced for most of the time, except when we age. As we age, the repressor of these retroelements become loose, and they start to slowly accumulate in your brain, creates a kind of a innate inflammation, chronic inflammation. But in space, we exacerbate that. So we create this inflammation really fast. And that is neurotoxic to these neurons. So then we postulate if Rett syndrome is caused, at least partially, by this neuroinflammation that makes the neurons not responding well, not working properly. If we block the expression of the retroelements, we should be able to revert the symptom. And that's exactly the experiments that we did. So now we are applying the knowledge that we gain in space to a condition here on Earth. And we start in all preclinical data, testing the animal, testing the brain organoids, using antiviral molecules that they are already like FDA approved molecules that people use to treat HIV for several years. So we know the safety procedure for these molecules, and it worked. And I said, oh, wow. I mean, who would imagine that an HIV drug is now being used to treat like a neurological condition? I mean, it was only revealed to the experiments in space. And I'm happy to say that we are moving forward with a clinical trial now. Hopefully, this will ameliorate the symptoms in Rett syndrome.
So you actually, that was going to be my next question, sort of like how we move from the things you were doing, which are more on seem to be more on the fundamental research side. how we're starting to translate some of this into actual therapies. Okay, so Rett syndrome seems to be like a very tangible example. Is there other things you're already working on or that you see on the horizon, like results of your research, which may result in actual therapies?
Yeah, so we are putting some effort on Alzheimer's now.
Wow. Okay.
And for Alzheimer's, we got Alzheimer's brain organoids. We already sent them and bring them back. And as we expected, we see several of these markers for Alzheimer's emerging quite early in the organoids that were exposed to space. And that includes markers of neuroinflammation, accumulation of beta amyloid, phospho-tau, neurodegeneration. So we see all those markers in the organoids. So the question now is, can we revert that? So we are testing some new molecules, and this is the part where I do it in collaboration with my colleagues in Brazil, where we are testing some of the molecules from the rainforest, from the biodiversity of the Amazon, to see if some of these molecules can actually revert. But we are also doing the experiments on prevention. If we pre-treat the organoids with those molecules, can we slow down the neurodegeneration? Are they protective? And that might actually end up helping NASA with future astronauts that might actually just take some of these natural elements before they go into space, so their brains are more protective. So Alzheimer's is one. ALS and Parkinson are others that we are slowly start to work with. It is hard to work with them all, right? I mean, it requires many people and many expertises, but that's how we are doing. Is slowly going to neurodegeneration.
Okay, interesting. And let me ask you about a completely different use case. We've been talking here about therapies for some horrible conditions, but I've seen at least once, I think it was a company in Switzerland, which is talking about using brain organoids basically as a biological substrate for computing. And that's obviously anything computing is a big topic at the moment because of artificial intelligence. And we're obviously using still very traditional chips and they're very energy inefficient. Is that something that has crossed your desk, sort of like thinking about using brain organoids for?
Oh, yeah. Together with a group of colleagues, I mean, we predicted that. So the current AI technology has several limitations. I think you point a few, like the cost, the training period for all this data that is used. So it's reaching like a plateau, right? I mean, there is no data enough in the world to train all these machines. So we are reaching a plateau. And then, I mean, the energy is a big problem. They are even thinking like putting like servers in the moon, or, I mean, we already have in our oceans. So brain organoids, because instead of artificial network, they have the real organic network. And the real organic network, it took millions of years of evolution to optimize that, something that we cannot copy. And nature find a way to store lots of memories, to create higher computational power in our brains with a very low cost. So how they do it, it's a big question, but we can already use it by using a brain organoid to execute some computational tasks. So we are calling that an organoid intelligence. And more I'm seeing companies. I am a founder of one of them that uses a brain organoid as a computational power to solve big problems in the world.
So that's super interesting. And I guess I have to ask from a sort of ethical review, ethics board point of view, is there going to be some limit? Right now you mentioned it's like a few million cells, so like these organoids are not, in a large scheme of things, they're not that big. But would there be some limit you see where we have to be kind of, I don't know, careful, for the lack of a better word, from an ethics point of view, where they kind of start becoming borderline sentient, or whatever the right word would be?
Yeah, it is a great question. And that's why, I mean, we team up with the Center for Bioethics here at UC San Diego. So we are constant in check with them. So all the time when we make like an advancement, before we publish, we discuss with them. We try to correct the right way to use the language, how to reach out to the best audience, exactly how to explaining the limitations and potential known anticipated uses of the technology. So we're always in constant check. And you're right. So at the time that this technology starts to become so sophisticated, where these networks might reach a level of sentient or consciousness, which, by the way, my personal opinion, it's inevitable. We're gonna get there. So it's gonna happen, right? But that might make some people uncomfortable. Some people might feel uncomfortable, especially the donors of the cells. They might not like this idea of having another brain of theirs sitting around in a lab making all these computational tasks. So that might create like an ethical or moral conflict. And what I'm doing is trying to create a discussion around that, so we are not taking the public by surprise, but we are preparing the public for some new technology that's coming. It is inevitable, and it's inevitable, as always happened before, that the technology was developed for something good and might be used for something bad. That's part of human nature. But if we can start thinking about ways of controlling that better, the earlier we start, I think the better.
Gotcha. And by the way, I forgot to ask in terms of use cases. So we talked about therapeutics. Okay, we talked about now computing. With some of the other organoids, including the ones we've flown to space, like, liver, for example, there's also been a lot of talk about use cases for regenerative medicine. So for example, from the liver that you could use organoids for liver regeneration. Is there any, so the brain is obviously more complicated to access and everything. Is there any regenerative medicine
Yes.
use cases of brain organoids as well?
Yeah, that's an excellent question. The brain organoids, different from like isolated cells, they are in a they kind of form the tissue, right? So you can imagine that, well, I mean, in conditions where we are losing brain tissue, you could eventually, from the same person, recreate that brain region that is missing and just transplant it back. So, and I'll give you one example that we did in a mouse model. So the animal, the mouse, the way it looks at things, it receives the light from the photoreceptors. It passes through the thalamus and reaches the cortex. And there is a visual cortex in the back of your head that actually creates the image that you are seeing. So what we did is the following: we opened the skull of the animal and we surgically remove the visual cortex from the brain. So the animal has everything intact except for the visual cortex. So it cannot process the information, the visual information. So the animal cannot see it again, even though their eyes are perfectly normal. All right. So once we establish that we create like an animal that cannot see it, and that happens in people as well. So if you have a concussion in your visual cortex, you might lose your vision. That's called cortical blindness, right? But then we take the human cortical organoid that we recreate in the lab and we transplant it back into the animal in the same region where we remove the mice tissue. And the cortical organoid fully integrate into the circuitry, become vascularized, start receiving input and output, and the animal starts seeing again. So now the animal are seeing using a network created by the human brain that was fully integrated into the mouse cortex. So it's an amazing example of the plasticity that these artificial tissues can have. So other applications that I think it's coming, for example, Parkinson, you degenerate your striatum that produces dopamine, and that's how you lose your movements. So can we create a striatum organoid and replace that from the patient? So I believe that's on the horizon as well.
Wow. So many amazing possibilities we already talked about. So Alzheimer's, Parkinson's, Reed syndrome, computing. So let me ask you this question. If you had unlimited funding and resources and postdocs and like what additional studies would you do?
So my lab is also, for example, interest on brain evolution. And that's how actually I started. My biggest question was always what makes the human brain so unique? What is unique about us compared to other species? And initially, I started doing like brain organoids from other primates, chimpanzees, gorillas, and compared to humans. But they were all very different. They were all like maturing really fast. By the time the human brain organoids is still migrating, the cells are still making the contact. The chimpanzee organoid is already fully mature. So there is no way for me to compare those two species. So then I had that idea, all right, what I need is another type of humans. And I started looking into the Neanderthals. But
Huh.
the Neanderthals, we don't have live cells from them.
Yes.
So then we decided to take a genomic approach. All right, let's swap the genes that are unique of humans, us modern humans, to the Neanderthal version. So in a way, we were using genome editing to fully Neanderthalize specific genes into a brain organoid. And by doing that, we're learning what are the importance of those genes during brain evolution. And one of them, it's called NOVA1, can actually, the Neanderthal version accelerates the maturation of the human brain. And in a way that it becomes more similar to a chimpanzee. And then you imagine why a Neanderthal version of the gene would do that. But then if you pay attention into evolution, a baby chimpanzee can outsmart a human baby. We are the exception. We are the ones that takes a long time to mature and to become independent. Even after we are born, someone needs to take care of us, so we don't know how to survive in the jungle alone, right? But the chimpanzee can do it. I mean, they were born, most of the babies in nature, they are born quite ready to fight, right? And to survive. So we are the exception, and that gene make us similar to a chimpanzee. So we believe that based on these data, that the Neanderthals are probably more close to a chimpanzee than us. And so if I have like unlimited resources, there are other hundreds of genes that we could study, and I'll do that.
And is there any interesting read across to some of this disease conditions we discussed? Because I vaguely remember that many animals actually don't even get Alzheimer's. But I don't know about
Right.
Neanderthals, but is there something there that maybe Neanderthal actually brain is better in some way, that it's more protected against diseases, and then it's something we can learn for the humans and therapies?
That's a great point, and that's exactly, I mean, the ultimate goal of the research is not only to learn about us, but how can we improve us. So our hypothesis is that by increasing the complexity of our brain during this very slow development, allowing the neurons to make way more synapses, way more connections than other animals, we also become, as a trade-off, become more vulnerable to some of these mental disorders or neurodegeneration. And you're right, there is no chimpanzee with Alzheimer's, there is no chimpanzee with autism. And as far as we can tell, those diseases doesn't happen in animals. So these are human specific conditions. And if we understand exactly how it helps, how it happens, we might be able to help people with these conditions. So, but you also touch like an intrigue proposition, which is maybe the other species also have something that are better than us. And I'll give you an example of the chimpanzee. They have such a great working memory that is like exponentially better than us. They can remember the sequence of numbers in a really fast way just by being exposed to a blink of an eye. While we're still figuring out what happens, the chimp already completed the task. So that's a remarkable ability to memorize things in connection with your visual input. And by far, outsmart us. Can we use that circuitry to somehow help us who have problem doing that? So that's another application.
So maybe for the computation, the biological computer is better on chimpanzee organoids.
It is possible. Yes, yes.
So this fast. So okay, let's come back to space, because so many of the interesting results have been from the micrograph from the space conditions research. So in my day job as a space venture capital investor, right, I interact with all of these people who are now proposing to build new space stations, right, like Axiom, Vast, and Starlab, and people who are doing free flyers like VADA. And so that's kind of, exciting in a way that there is, so far we basically principally had the International Space Station, and that's obviously, it's great. And now also there's the Chinese space station, but it looks like within a few years we could have many platforms to conduct research in space, which should be very exciting for researchers like you, I guess. Is there something like a wish list you have, like while these platforms are being designed? Like, is there something we like a message you would leave to these companies, like, oh, please do that, or please design the station like this, or please have this equipment, or something that would help your work?
Yeah, we are already in touch with most of them, so they are aware of our work, and we've been, for some of them, providing advice. on where the biology is going. So most likely they will need some kind of stem cell lab production in space. So instead of us keep sending organoids back and forth all the time, why not just growing in there? So then we would have like a facility that will grow the organoids and send it back to Earth. So we can perform our tests here, but they already are coming with a more age phenotype, so we don't have to worry about that. So that's definitely something that we are discussing with some of them.
Okay. Is there any particularly equipment you think we should have in space that we don't have yet? I have some people saying, for example, oh, we don't have good DNA synthesizer. We should have that in space. Or I don't know, you're a brain researcher. Maybe you want to have like a functional MRI or I don't know. Is there any equipment missing in space?
So the only equipment that we're missing, we're working on it, and it is a way to do electrophysiology in neurons in space, because most of the technology require gravity for the neurons to attach to an
Hmm.
electrode. But we have like a prototype that seems it might work in the way we think. But more important than equipment, I'll tell you the limitation. The
Mm.
limitation is the scientist.
Mm.
And because the type of science that we do here, there is no astronaut that can reproduce that. So
Mm.
you need to be a professional to do it. So it's almost asking like, I don't know, a mechanical guy or a handyman to do like a neurosurgery. So it's not going to work just by teaching them how to do it or explaining today over a year. So that requires years of training for you to be good at that and be confident to perform that experiment. So we have the following proposition for the community is to create a cohort of scientists that were trained to become an astronaut. And of course, I mean, since these are professional scientists and they're not gonna go there to do like maintenance of the space station.
Hmm.
So that's
Hmm.
not the point. And they're gonna go there to do exactly the experiments that they were trained for. So this would be like a new workforce, a new category of PhD students, postdocs, that needs to be trained both on Earth and in space to perform these experiments.
Okay. So that kind of partly goes to my next question I want to ask, sort of like, if you look like, let's say, 10 years out, where do you see sort of in space research and experimentation, or where would you like to see it, I guess?
Yeah, I would
I guess part
like
of
to
that is
see.
that scientist cohort you were talking about.
Yeah, exactly. I would like to see us in the lab here taking turns to go to the space station and perform the experiments. So one week, one of us will go and prepare the experiments. In another week, another one will go and perform the experiments. In another week, someone will bring them back. I see like I imagine like a very dynamic group of people going there and preparing these experiments, bringing back to Earth, having results here on Earth, working more collaborative with industry,
Hmm.
getting whatever drugs they have on the shelf. Let's test that for Alzheimer's. Let's test that for Parkinson's. So accelerating drug discovery by have a more relevant human model to
Hmm.
study with.
And so that now is a nice segue to my next question. And you mentioned industry there, and so I'm hugely optimistic, as you can probably tell, about the intersection of space and biology. But I've been trying to have conversations with big life sciences pharma companies, with specialized biotech investors, and there's still a lot of, let's call it, hesitance or prejudice. I mean, very often, I don't know about you, but very often I get the reactions of like, space, like really, no, like why? That like is complicated, expensive, only for governments. Is there I mean, A, are you seeing the same thing? And B, if you're seeing the same thing, like how do you think we can fix that? Or is there like a message you want to leave to life sciences companies that may
Yeah.
be listening?
I and I understand the reluctant because we don't have that many examples,
Hmm.
especially on biology. So how many drugs did we discover in space that we are actually using here on Earth?
Probably some osteoporosis drugs for memory.
Exactly. The Mgen compound was discovered in space to avoid like bones to break, and now it's the osteoporosis drug. But that's it, right? So there are some people using like crystallization in space,
Mhm.
make the manufacture a little bit better than on Earth. That's great, but not many examples. So I think as soon as we start showing that, I mean, we can actually find a new treatment for Alzheimer's. It might be on my my lab right now. So then this idea that, oh, these are expensive experiments. No, I mean, how much we invest so far on Alzheimer's? We're talking about billions of dollars,
Yes.
zero return
No
of
results,
investment.
basically. Yeah.
Yeah.
Yes.
So maybe like 1 or 2 trips to space, we might find it. So that doesn't become that expensive anymore. It becomes actually very practical to do that. So I think what is missing is examples. And I'm hoping that this will be part of my contribution to science, is to show examples like the one I mentioned about Rett syndrome. Having a treatment for this neurological condition. Again, I've been studying it for 20 years and there is nothing there. I mean, people are thinking about gene therapy, but there's still like some years to go and might not work. So we need to be prepared for something. I think this would create like more excitement in this field. And then the whole ecosystem. When I started, not many rockets going to the
Mm.
International Space Station. So we have to wait, fight for a spot there. But over the years, especially due to SpaceX activities,
Yes.
more and more rockets are going there. So we are having more and more opportunities. So it's improving over time. Let's hope that NASA will continue to support these activities in the current situation and we don't drop the ball because
Yes.
this is really innovative for US to be like in the forefront of this biomedical research.
No, I fully agree. So I certainly hope the ISS National Lab is going to continue its great work. But then, like, speaking of SpaceX, I mean, there was, just a few days ago, news about their Starfall project and that they may get into more biological research themselves. So yeah, hopefully the number of rocket flights, the number of opportunities to research is going to further increase. So we could go on for hours. I'm so fascinated with this topic, but we are slowly running out of time here. So if people are listeners listen to this and are interested, is there any sort of easy places where they can get more information about what's going on in this line
Yeah,
of research?
I mean, I'm open to collaboration to discuss, and people can easily find my email on the internet. You are welcome to come here and visit the lab and tour the facilities.
Certainly will.
And as soon as we start having the publications, I think people would start downloading our data and start seeing exactly what is done. So the pace of research in space is different from the experiments on Earth, right? I mean, we basically do one experiment per year. So it takes time to have like a decent publication because you want repetition, you want to validate what we found in one mission in another one. So that's part of the game too. I mean, if you're too anxious about having publications, this is not for you. So you should be prepared for that.
Understood. Alysson, the last question in this podcast is always the same, independent of the guest. It's about science fiction and, whether you like science fiction. And if you do like science fiction, then if you have any favorites you would like to recommend.
Yeah, I love science fiction. I always get inspired by my favorite. It is Blade Runner.
Okay, very, very, very good answer. Well, listen, thank you so much for coming on to talk about your fascinating research, and we're really looking forward to more published results. And yeah, best of luck with everything you have going on.
Thank you so much. That was a great talk. Thank you. Yeah.
And that's it for another nominal episode of the Space Business Podcast. And once more, if you enjoyed this episode, please leave us a 5-star rating on your favorite podcast platform, such as Apple or Spotify. You can follow us on Twitter at podcast_space. We're also on LinkedIn under the Space Business Podcast. Lastly, if you have any feedback, including ideas for guests, and that may even include yourself if you have an exciting space story to tell, or interested in becoming a sponsor, or really for any other suggestions, you can drop us an email at spacebusinesspodcast@gmail.com. Thanks so much for listening, and see you for the next episode.